So we're going to be picking up on Chapter 2 again today, but before we get back into it, I just want to ask, are there any questions that people have about the material that we've talked about so far? Okay. I know that there's been a fair bit of kind of overlap with previous topics so far, and it will continue to get more and more dense with material you haven't seen before. But the stuff that I'm talking about now is really important. And so if you're at all fuzzy on it, it's real worth getting on top of it and making sure you have a handle on it. I did talk about this slide a little bit on Monday, but I didn't point out the examples of hydrogen bonds on it, just sort of trying to remind you that a water molecule here can, through its partial positive of the hydrogen, bond to an alcohol, the oxygen of that alcohol. But it can also bond through the oxygen of the water to the hydrogen of an amine, for instance. So the water molecule can be either a donor or a recipient on a hydrogen bond. And because there's so many polar bonds in molecules and cells, there's lots of things that can interact with water molecules. So what makes water such an extraordinarily good solvent and such a, therefore, a wonderful medium for living systems is the fact that it's a very broad spectrum solvent. And it's such a remarkably good solvent precisely because of this polar character it has, where not only its own bonds are polar, but the molecule as a whole is polar. And these are just two examples of the kinds of things that water does acting as a solvent. So here you've got a sodium ion and a chloride ion. These were originally presumably together as a salt, and water was very good at separating them out of that crystal because the water could surround the positively charged sodium ion with the partial negatives of its oxygens, and then surround the chloride with the partial positives of its hydrogens. And so now these two ions have a little shell of water around them that acts like a little bumper almost. Water is also very good at interacting with polar compounds, and this is just an example of glucose. You can see all these oxygens in a glucose molecule, and so therefore all the potential sites for hydrogen bonding either to the oxygen or to the hydrogens of the alcohols. So obviously glucose is a really good compound to dissolve in water. That's part of, but not the only reason why it ended up as the principal energy source for us. So we've talked about water as a polar hydrogen bonding compound and why it's such a great solvent. It makes a texture of a medium where each molecule is extensively hydrogen bonded, at least 3, often 4 to each other, and wonderful at surrounding polar things. But there are some things that aren't polar. And now we get to talk about the hydrophobic effect. Hydrophobic literally means water-fearing, like arachnophobia, who's fear of spiders. The parallel adjective for things that are polar would be hydrophilic, loving water, a little bit projecting our feelings onto things here. But in general, nonpolar compounds, and the example that we're showing here is the example of the hydrocarbon dodecane. So this is a string of 12 carbon atoms, and all the other available bonds are filled with a hydrogen. So carbon and hydrogen are fairly close. A carbon-hydrogen bond is not particularly polar. This molecule as a whole has no very remarkable locations where it has a partial positive or partial negative charge. So it's nonpolar. And we call it a hydrophobic molecule because it's going to interact with water in a different way than the polar ones. With a polar compound, the water molecules can tumble and poke their partial positives or poke their partial negatives at those polar areas. This compound doesn't have either one. So the water has to, in effect, make a little space for it to fit inside of where it's not orienting its charged areas towards it. And so as a consequence, there is a loss of entropy in the area around a nonpolar compound. So here we have our lentil bean-shaped nonpolar compound. We're putting it into water. And over here, we can see this kind of layer around that nonpolar molecule where those water molecules are really not free to interact with it the way they would with each other or they would with a polar compound. So it's a patch, if you like, of order, of reduced disorder, of reduced entropy. It's a loss of entropy, a loss of disorder, just by putting a molecule into the solvent. Now, this has an interesting consequence. If you put several of those nonpolar compounds into the water, there are several of them in there, they will tend to gather together. And that gathering together or aggregation is, we say it's driven by the hydrophobic effect. And diagrammatically, it would look like this. You've got each of your little nonpolar compounds. Each of them has their own little shell of water that's not really free to do what it would like to do. If all of those nonpolar compounds collide and hang around with each other, the area that has to be constrained like that is smaller. We've reduced the surface of constrained water. We've created a little bit of order by bringing together the nonpolar compounds, but the disorder, by permitting more of the water solvent to go around banging and switching and stuff, will more than pay for that. So the hydrophobic effect is not that there's an attraction between nonpolar compounds. There isn't any particular attraction. It's more like imagining a big mixer where there's 4 or 5 people who have very few social skills, and people don't really stop to talk to them very long, and they end up in the corner together. It's not because they're attracted to each other. They've just kind of collided there, and everybody else has left them alone. So this hydrophobic effect turns out to be very powerful. And it turns out to underlie a very large fraction of structure formation in biological systems. So the hydrophobic effect we're going to see accounts for the formation of membranes. The hydrophobic effect is one of the driving forces in the assembly of many proteins. It contributes some to nucleic acids, but nucleic acids also have stacking interactions which are a little different. What we're saying is that the 3 classes of macromolecules we've talked about, 3 of the 4 classes of molecules have important, significant hydrophobic interactions. And we're going to expand on all of these as we get to each of these as we discuss structure. So we've talked about hydrophobic compounds that are nonpolar and don't really interact with water. We've talked about hydrophilic compounds that are polar and love to interact with water. If you've ever tried to make hard candy, you know how much glucose you can get into water, right? Suppose you had a compound that part of it was polar and part of it was nonpolar. Well, then you'd have an amphiphilic compound. And amphiphilic is just Greek, like a lot of science. It means loving both. Like an amphibian lives on both land and water. Amphiphilic compounds love both the polar parts and the nonpolar parts. And this is an example of one typical amphiphilic compound. The polar head here, you can see those carbon atoms. So this is a carboxylic acid, is polar. Because it's polar, it's hydrophilic. Excuse me. It's hydrophilic. The other end, which looks very much like that dodecane, is just a long wiggly chain of carbons with hydrogens covering over it. This part is nonpolar, and because it's nonpolar, it's hydrophobic. So it certainly is possible to have molecules that are amphiphilic, and ones that have very interesting properties. Since part of the molecule likes to interact electrostatically with water through the polar parts, and the other part tends not to interact with it, and if given its chance would pull away from water or be shunted out of water, these molecules take part in a sort of a self-assembly process. And the typical thing that happens is that the nonpolar tails sort out of water. And these are very common ways of diagramming these amphiphilic compounds. The reason that the nonpolar tails are sort of shown as squiggles is because those hydrocarbon tails have very few constraints on their structures, and they can flap out extended or they can curl up. I mean, they're just kind of dancing around like spaghetti. They're not very structured at all. Whereas their polar head groups tend to have more specific structures to them. And they can sort out from water in a couple of different ways. They can make lipid bilayers. So this is a bilayer because there's 2 layers of lipid that have organized relative to each other. Or they can sort out as a micelle. And in both cases, the polar head groups are in contact with the water. And in both groups, the nonpolar tails, the hydrophobic tails, are spontaneously sorted away. No one had to tell it where to go. But the shapes are quite different. And you can see this one would make in a sheet, an extended flat sheet, and this makes like a little teensy tiny droplet. And the difference lies in the architecture of the molecule. So in a group like this that has two nonpolar tails, the space it takes to fit both of those tails in is such that it can pack into a lipid bilayer like this, but it could not pack much tighter than that. Whereas in the case of a group like this that has a polar head group and a nonpolar tail, that polar head group is now, relatively speaking, probably bulkier than the tail, and the only way to fit all that together is in a micelle like this, where those head groups are in contact with the water, and then the tails just kind of smoosh around in the middle. These are going to turn out to be important as we talk about biological structures. Any questions so far? Okay, great. Now, what's very useful about lipid bilayers is that because the core of the bilayer is hydrophobic, it's nonpolar, it's hydrophobic, it will prevent the diffusion of polar compounds. That really accounts for a surprising fraction of compounds. So salts are obviously going to be polar. Proteins in general have polar regions or have a polar surface. Many metabolites are going to have polar aspects of that. So what that means is that we can preserve or hold on to things by surrounding them with a lipid bilayer. The tendency to diffuse follows the law of mass action. That is, you go from a more concentrated to a less concentrated. So in the absence of anything else, if you had a little pile of metabolites and proteins just there in the water, it would diffuse away into soup. If you were to wrap that around with a lipid bilayer completely surrounding it, it will stay together. Clearly, if the mechanism of action is that the lipid bilayer has a nonpolar interior, and that's what holds things in, we can make predictions about what things can and can't go through that lipid bilayer. What about nitrogen gas? Would you predict it would diffuse through a membrane, or would it be retained? Sorry? Diffuse. Great. Anyone want to volunteer an explanation for why it would diffuse? It is small. Small will certainly help. It's nonpolar, right. So if you look at the molecule, it's two nitrogen atoms. They're the same electronegativity. The bond is nonpolar. So we could look at O2 and have the same kind of argument that oxygen can diffuse through it because it's essentially a nonpolar compound. This is one of the reasons you can't get much oxygen into water, is because it's a nonpolar compound. Water. Can water freely go through a lipid bilayer? Any brave souls? It certainly is polar, and you would predict from its polar character that it should not go through freely. It's small, and some people thought because it's small it should go through. And if you looked at the behavior of cells, it looked sort of like cells were behaving as if it could pass through the lipid bilayer. We found out recently that there are actually pores in membranes called aquaporins, so that the polar character of a water molecule is sufficient to prevent it from simply diffusing through a lipid bilayer. Because our cells, everybody's cells, have this lipid bilayer around them, we're able to maintain specific ionic compositions. And it turns out that this ionic composition is relevant to how we go about doing the business of living. And in general, we can say things about the interiors of cells that hold true for almost all cells, which in its own right is an interesting fact. The intracellular medium of a cell typically is going to be enriched for potassium. The extracellular materials, if you have a cell that has a body as a whole, so it has an extracellular fluid that's part of the organism, our extracellular fluid, and that of most animals and most plants, has sodium and chloride in it, not too dissimilar to the composition of the ocean. And because the lipid bilayer holds these apart, cells can maintain a difference there. Look at the intracellular composition. So this is a positive charge, that's a positive charge, that's a negative charge. there's a whole lot more positive charges there than negative charges. Any notions on what balances this electrically inside of a cell? We mentioned that nucleic acids have a lot of negative charges on them. Turns out a lot of other metabolites have a lot of negative charges on them. So in general, biological systems have enough negatively charged biomolecules to counterbalance that charge. Okay. Sorry. So far we've talked about the water molecule as if it stayed together forever, but it doesn't. Not quite. The fact that the water molecule can dissociate is the basis for acid-base chemistry of aqueous systems. There's a whole different set of acid-base chemistry for non-aqueous systems about which I am massively ignorant. When water dissociates, we can think of that as dissociating into a hydroxide ion, OH, and for convenience, a proton. In practice, it's more likely to be a hydronium ion most of the time, but we'll usually talk about it as a proton, so a positive and a negative charge. And because water molecules have multiple sites for hydrogen bonding and so on, they also have multiple sites where that proton can go on and off. And you can imagine this movement of protons. If this hydronium ion loses this proton, it might jump to here. This proton might fall off and go over there, and so on. So in a liquid medium of water, the protons are bouncing off and jumping back on with extreme rapidity. They're not just simply sitting there like someone waiting to be picked up. Okay. So water can dissociate, but it doesn't do it much. Not at all. The dissociation constant for compounds is described as a constant K, with the concentration of the dissociated products on the top and the concentration of the undissociated original stuff on the bottom. Now, because water does not dissociate very much, we can treat this bottom term as a constant. And so the water dissociation constant can be simplified into this term of H, the concentration of the protons, times the concentration of the hydroxide ions, and we can say overall, that product of their concentrations will be a constant. If you have more protons, there'll be fewer hydroxide ions to make sure it stays constant. And that water constant is really small, 10 to the minus 14th. So at neutrality, you have 10 to the minus 7th molar protons and 10 to the minus 7th molar hydroxide ions. You don't have very much of either one of them. That would be neutrality. And because the dissociation constant is a constant, if you have a higher concentration of protons, you're going to have a lower concentration of hydroxide ions. If you have a high concentration of hydroxide ions, you're going to have a lower concentration of protons. So, it is such a huge range over which water can do this that quite a while ago, chemists and biochemists agreed to a different way of handling it called pH. When you have 14 orders of magnitude, you need a tool. And so the tool that we have is called pH, in which pH is the negative logarithm of the concentration of the protons. So concentration of protons will be in molar. The negative logarithm means that when you talk about 10 to the minus 7th molar, that would turn into a pH of 7. So at neutrality, when you have equal concentrations of both protons and hydroxyl ions, the pH will be 7. And this scale here shows you the whole spectrum, both in the pH scale here and in the concentration of protons over here. So pH 7 has 10 to the minus 7th molar hydroxyl protons. pH 2 has 10 to the minus 2 molar hydroxide ions, and teensy-tiny amounts up in there. This allows us then to talk about all solutions, all aqueous solutions, as being on one side or the other of neutrality, where you have equal amounts of protons and hydroxide ions. At 7, we have neutrality. If pH is smaller than 7, that will be acidic. And the smaller the number, the more acid the solution is. If it's higher than 7, it'll be basic, and the larger the number, the more basic it is. And this is not new to you, but sometimes people aren't motivated the first time they see it. It will turn out to be important. So we can change the solution of pH we can change the pH of a solution. If you add acid, it will lower the pH by raising the concentration of protons. If we add base, it will raise the pH. Here we're working with the definition of acid as a proton donor and a base as a proton acceptor. So if you have hydrochloric acid and water, when you add this, which is a gas, to the water, it will donate a proton to make hydronium ion and a chloride ion. Here's sodium hydroxide, which is a base. If we have a solution in which there are protons available, when this dissociates, that hydroxyl ion that it donates will pick up the proton, and now we'll have a sodium ion and 2 water molecules. So you might want to work out these details on your own, since I'm looking at the time. Now, we've talked about the water dissociation constant. It turns out, by the way, there may be a duplicate slide in your, not yet. Don't worry. I think this is it. This is okay. I want to be a little careful here. Okay. All right. Now, a lot of the compounds that are present in biological systems are what we call weak acids or weak bases. That is, some of them will dissociate, some of them won't. If you put hydrochloric acid into water, it will dissociate. If you put sodium hydroxide in, it will dissociate. Most biological compounds, some will, some won't. And so we need a number that will describe the tendency for those things to happen. And here we're looking at acetic acid in water. So the reaction that we're thinking about is acetic acid in water can go to hydronium ion or proton and acetate. Acetate is the ionized form. And so the dissociation reaction is described this way, with the dissociation products on the top and the starting material on the bottom. Under any conceivable circumstance that we would do this, the concentration of water won't change. So we can treat water as if it were a constant. And then we can have what we call a Ka, an acid dissociation constant. And this is the product of the dissociation products over the starting material, the acetic acid. So this is a way of talking about its tendency to dissociate some, not a huge amount, just as we did for the water dissociation constant. And because the kinds of concentrations that we deal with are themselves these large orders of magnitude difference, just as we developed pH as a way to talk about acid concentrations or proton concentrations across a huge spectrum, the terminology or the method of describing it as pK was developed. So pK is going to measure the tendency for weak acids and bases to dissociate. And pKa, or the P of the acid dissociation constant, is the negative log of the Ka. And in the case of acetic acid, the pKa is 4.76. If it has a large Ka, so if we just go back to this one, if K here was a large number, that would mean there was a lot of dissociation, right? So with a large Ka, you'd have a lot of dissociation into the ionized products. Because this is a negative logarithm, you would have a smaller pKa. So the smaller the pKa, the stronger the acid. And this, I don't expect you to memorize these numbers. This is just to give you a sense of the spectrum of pKa's and a few somewhat important examples. So we've just mentioned that acetic acid has a pKa of 4.76. Some of you may work with buffers called ACEs or imidazole, which have pKa's here, you can see are very close to neutrality, 6.9, 7. So these protons, the ones that have been highlighted in red, which may not be too visible on your handout, but those particular protons have almost the exact same tendency to dissociate that water itself does. HEPI's not quite so much, it's a little higher. Ammonium ion, rather high pKa, so it's going to tend to behave as a base. Okay. This, I think, is the place where there may be duplicate slides. And let me just see what got into the handout. So I think you want to look at the one that is called slide 27. I deleted the duplicate. So. When a weak acid and a weak base dissociate, they can affect the pH of a solution. And their tendency to affect the pH of the solution will depend on how likely they are to dissociate. And we can talk about that in a quantitative way by looking at those relationships. Let's look at this weak acid. So it's HA. Could be acetic acid, could be something else. Some weak acid is capable of dissociating into the conjugate base and a proton. Conjugate base can receive a proton, which makes it a base. We can talk about the Ka for that compound by looking at the dissociated products over the starting material here. We can rearrange that equation so that things are in different places. And if you're curious to see that rearrangement, stop by afterwards. But we can rearrange this equation so that the proton concentration is on this side. And we can now relate the proton concentration to the acid dissociation constant, the concentration of the weak acid, and the concentration of the weak base. But we don't work with proton concentrations routinely. We work with pH routinely, and we work with pKa's routinely. So we need to convert this equation to negative logarithms. So the negative logarithm of the proton concentration will equal the negative logarithm of the Ka minus the logarithm of the concentration. This is a little bit confusing. Minus the logarithm of that ratio. So that the pH of the solution can be predicted by the pKa, the tendency of that compound to dissociate, and the ratio of the weak acid to the base. I'm not going to ask you to calculate any solutions to things, but I want you to have a sense of qualitatively what this equation is trying to tell you. It's trying to tell you something about the tendency of a group to dissociate, and the presence in the solution of the other compounds will change the pH or not. One of the things that makes pH and pKa and the ionization of biological groups and so forth a little bit frustrating for some people is because when we're talking about ionization of functional groups, which word is used at which time is going to depend on what kind of a group we're looking at. And so I'd like to walk through with you just for a moment a couple of the groups we're going to see very frequently through the rest of the semester so that you're comfortable also with what happens when. So if we think about three different possible conditions, we can think about when the pH is less than 2. This is a proton-rich condition or an acid condition. We can think about an intermediate pH regime, which is on sort of flanking neutrality. Or we can think about pH greater than 11, which has few protons around, or we would often call it a base solution. In an acid-rich environment like this, a carboxylic acid like acetic acid, for instance, there's lots and lots of protons around. This is below the pKa of this group. It's going to tend to be protonated. It's picked up a proton here. It's in a protonated form. Ammonia is going to be protonated. This is a proton-rich environment. Both the groups are protonated. As we continue to raise the pH, and there are fewer and fewer protons around, the carboxylic acid will probably have lost its proton by the time you get into that neutral regime. But the pKa for this amine group is quite a bit higher, perhaps around 9. You remember, we looked at the ammonia. So it's going to still be protonated here, even though there are fewer protons around than there were before. You get to the very higher pHs where you have very, very few protons around, and finally it's going to have lost its proton as well. But notice how differently that affects their ionization. All right? At the low pH, they're both protonated because there's lots of protons around. At the high pH, they're both deprotonated because there aren't very many protons around, and when they dissociate, they don't pick one back up. But the ionization depends on the group that we're talking about. This is an ionized group. It has a negative charge. So for the carboxylic acid, it becomes deprotonated as it becomes ionized. By contrast, if we look at the amine group, here we are. It's protonated because this is a proton-rich environment. We're raising the pH, and eventually it gets deprotonated. It is unionized. It is no longer charged. So protonated and deprotonated refer to losing or gaining the proton. Ionized or unionized is going to refer to charged or uncharged because you've lost or gained one. But they are not going to be the same for all groups. Sometimes protonating something deionizes it. Sometimes deprotonating it deionizes it. You know, it's going to depend on the group you're talking about. And if this is in the least bit fuzzy for you, you need to get comfy with it before we go further. The fact that weak acids and weak bases tend to dissociate but don't just fall apart allows us and allows cells, more importantly, and organisms, to use these weak acids and bases to perform a very important function called buffering. If you were to look at what happens to the pH when you add a strong acid like hydrochloric acid to water, you add the hydrochloric acid, it dissociates to HC, H plus, and Cl minus. You've got lots and lots of H pluses. Whatever amount of hydrochloric acid you put in, you've got that many protons now in solution, basically, fundamentally. So you get a large change in pH for adding your strong acid. If, on the other hand, that solution had in it a weak base in there, remember we said that the conjugate base to a weak acid is a weak base. So here's A minus. This is the conjugate base, might be acetate, something like that. Okay, the hydrochloric acid goes into the water. There's lots of water around. It dissociates into the protons and the chloride ions. But this is around to pick up some of those protons. And so if you have the conjugate base around, when you add a strong acid, you're not going to have the full change in pH that you would have had otherwise. And so the presence of this compound is going to reduce the change in the pH. We say it has buffered the change in pH. It is taking an input and making it have a smaller effect. And this is just a diagram for acetic acid and acetate. And this has on it the examples of what I was talking about being protonated or ionized and the difference between those things. So if we start off with if we were to look at a solution that's down around, a little below 3 or something, we have a solution that has a large number of protons free and available, and so that's going to tend to favor the protonated form of the acetic acid like this. If we start adding base to this solution, we can add sodium hydroxide, for example. We can add base to this solution, and that would normally be raising the pH because it would pluck up those protons from the water molecules. But as you add that base to titrate up, you're going to start seeing more of the deprotonated forms formed because you're changing the pH. But you're not going to see a linear increase in pH. You're going to see a more gradual increase. And that change in pH will have an inflection point at some point, and then the pH will start to rise again until you get to the end of it all. This inflection point at the middle, this midpoint, has a very interesting property. At this midpoint, there are equal numbers of ionized and unionized acetate there, or acetic acid acetate, equal quantities of those things, equal amounts of protonated and unprotonated forms. And as you continue to get past the midpoint, you're going to get more and more of the deprotonated forms, or the acetate. Okay? You could start at a high pH and titrate down. This is insensitive to which direction you're going to. But there's a range here in the middle where for a substantial addition of base or acid, there's a small change in pH. And we call this the buffering range. And this midpoint turns out to be equal to the pH. The pH of that midpoint on this titration curve is the same as the pKa for that weak acid or base. And so that allows these weak acids and bases to serve to buffer changes in pH as there's production of protons or consumption of protons. Any questions about that before we move on? Okay. Acid-base balance is extremely important in humans. I've always been struck by Cannon's comment that the human being is delicately balanced between convulsions and coma. And acid-base balance is one of the things that does that. In our bloodstream, we have a substantial amount of bicarbonate ion. You've heard of sodium bicarbonate, right? Sodium bicarbonate is bicarbonate ion plus a sodium ion. Well, the bicarbonate ion in the blood has the interesting property that it can pick up a proton to become carbonic acid. And this carbonic acid can then further rearrange and dissociate into one molecule of water and one molecule of CO2. So that's, under normal conditions, there's a certain amount of each of those things in the bloodstream, and we're merry and we're not in convulsions or in a coma. If there's an excess production of acid somewhere in the body, so for instance you're running very hard and you're producing acid in the periphery or something, the pH, the protons that are being released by that metabolic process out into the bloodstream, when they encounter the bicarbonate ion, you're going to tend to have, by the laws of mass action, more carbonic acid formed. And if we make more carbonic acid, it's going to tend to dissociate into carbon dioxide and water more. And then that carbon dioxide can be breathed out. And so you breathe it out. If you have not enough acid in the bloodstream, the other direction can also help in restoring that balance. That you can you breathe in a certain amount of carbon dioxide from the outside, it forms more carbonic acid, and then that carbonic acid dissociates to restore enough protons for the bloodstream. If any of you know people who've gotten very, very anxious, one of the things that happens when you get very anxious is you tend to breathe in a sort of rapid way, more rapidly than you really need to get your oxygen. And you could end up blowing off, they say, some of the carbon dioxide that you need to keep that balance in the right range. And so one of the things that can help under those conditions is to get a paper bag and just rebreathe that air. And what you're doing is you're raising the carbon dioxide concentration inside the bag and then eventually raising it in your blood and eventually restoring the pH balance in the bloodstream. So, questions about that? This is a very important part of kidney disease. Maintaining the proper balance of all these ions can lead to bad things happening when you have kidney disorders too. Okay. The last few slides are a chance to just look at or apply some of the things that we've talked about in specific ways. So for instance, take a moment and look at your uric acid and talk to each other and take a look at this molecule and ask yourselves, does this compound have on it hydrogen bond donors or hydrogen bond acceptors? Okay, sort of pick someone near you to chat with for a second and sort that out. Okay. Do you have? You don't? Did she not include this in your? Okay, there you go. There you go. Okay. anyone want to care to venture how many hydrogen bond donors, if any, that uric acid has? How many? Any idea? Anyone? Where would we expect to see a hydrogen bond donor? Yes. So this nitrogen here and this nitrogen, right? In fact, I think basically we could expect all of those nitrogens to have a somewhat polar bond and be able to be hydrogen bond donors. Wouldn't you say, Eli? Yeah. So we've got 1, 2, 3, 4 hydrogen bond donors, and therefore how many hydrogen bond acceptors? A little louder. 3, right. Which would be which atoms? Right. So each of these oxygens that are on a carbonyl carbon, each of those could accept a hydrogen bond. Uric acid is a pretty soluble compound. You can do this kind of analysis for all of them. Take a look at these compounds. We've got 5 compounds here. And they're asking, do intermolecular bonds form? So we have intermolecular bonds in water that make water a medium, that make it a solvent. So for each of these 5 combinations, we're going to ask, do intermolecular bonds form? And if so, where do they form? Okay, so in this case, there's just one compound, one compound, one compound. Here we have 2 compounds, and here we have 2 compounds. So give that a little look-see. You may not have had time to discuss all 5 combinations yet, but let's work through at least a couple of them and then see if we want to get on to another one. For compound A, do you think that that compound can form intermolecular bonds with other molecules of that compound? Yes or no? Does somebody who wants to say yes want to raise their hand and say why? Or somebody wants to say no, say why? Yes.
Carbonyl carbon is partially positive and the oxygen is partially negative.
Right. So you can be involved in interactions like that. Now, what's interesting about the fact that you've got the partial negative on there and the partial positive on the hydrogen and carbon is that you have one of each. And so you can imagine making chains of this molecule, but you can't make a network the way you do with water, right? And so you would predict on first principles that this would not have as high a surface tension as water. It would not have as high a boiling point as water, and so on. So just looking at the structure of it, we can make certain predictions about it. How about this compound? Yes? Okay. You want to take a stab at explaining your yes? So you can make a hydrogen bond where this might be the hydrogen bond donor and that would be the hydrogen bond acceptor. And again, you get that kind of linearity. If you look at this compound, this is methane. This is ethanol, right? This is ethanol. And ethanol is, in fact, a liquid at room temperature. But it has a lower boiling point than water. And it has a lower surface tension than water. So, okay. Let's take a look at... Let's jump ahead a couple more. We may come back to that if we have time. Anybody recognize this name? Some of you have had lab courses. You've probably seen it in lab courses. Have you seen it anywhere else? Sorry? It's shampoo, right. So the cheap shampoos will say that they contain sodium dodecyl sulfate. The expensive shampoos will say they contain sodium lauryl sulfate. They're the same thing. It's just you pay more for sodium lauryl sulfate. Is this compound polar or nonpolar? It's both. Great. Which parts are which? Right. So this chain here is going to be nonpolar, and this part of it's going to be the polar part. So we have an amphiphilic compound. And so why do you think it gets your hair clean? Yeah?
The non-polar part invests itself into the dirt particles, and then the polar part's the away by the water.
Right. So a lot of the dirt that's in your hair is going to be the oil and stuff on it, and that's just going to get mixed into there. And there are some polar parts of dirt, like salt, like sweat is a polar part of dirt. But one way or the other, it can interact and then pull it into micelles, typically in the water solution. And we're actually fairly close to the end of the hour, so I think I'll say see you on Friday.